So you can see, you can memorize the important intermediates you already know. So citrate, actually citrate will form fatty acid. Citrate will form fatty acid. We will talk in fatty acid cycle. So what happens? This pyruvate, you know, pyruvate converts into acetyl coenzyme A in what? Mitochondria. This acetyl coenzyme A with oxaloacetate, it binds and it will form citrate. And this citrate will actually go out of the cell through pathway called, this is a transporter called citrate shuttle. This transport system is shuttle system. So transport, and this citrate goes into the, this is cytosol citrate, and this citrate will again break into this acetyl coenzyme here, and oxaloacetate will come here, not as oxaloacetate, I have directly shown as oxaloacetate. So it will come like this. Now this acetyl coenzyme A will form fatty acid. So citrate will, citrate is connected to fatty acid synthesis. How is it connected to fatty? What will happen? This cycle will actually goes on until ATP become high. When you have taken very high amount of fat, ATP and NADH become very high. So this will, the pathway will stop, and then this citrate will divert for formation of fatty acid. So citrate is involved in fatty acid synthesis. Remember, citrate, fatty acid synthesis, very important. Succinyl coenzyme A, we will talk about this also. It is a very important element of heme synthesis. It is substrate for heme synthesis. Succinyl coenzyme A plus glycine will combine and it will form heme. So remember, heme synthesis. Fumarate, we will talk about this heme synthesis, we will talk in MD1 only. This will also, we will talk in MD1 only. But fumarate is required, actually is part of urea cycle. So urea cycle we will talk in MD2. So remember, fumarate is connected, urea cycle is connected with fumarate to Kreb cycle. So urea cycle or Kreb cycle are connected. Function as gluconeogenetic substrate. So malate will form glucose. These are glucose. So we will talk about how Kreb cycle help in glucose synthesis. We have that. So these two we will talk little bit now. So these are the connecting pathway, significance of this Kreb cycle, because I have told you Kreb cycle is an amphibolic pathway. Amphibolic pathway. So this Kreb cycle, in fed condition, it will burn glucose. It will burn glucose and produce CO2. But this happens in fed condition. But in fasting condition, what will happen? This Kreb cycle will produce glucose. In Krebs, in fed condition, it will burn glucose and produce fatty acid in fed condition. In fasting condition, it will produce glucose. So glucose will be produced. We will see how glucose is produced during fasting condition in a very brief. Kreb cycle is utilized for gluconeogenesis. I told you it is required for glucose synthesis as well as for glucose breakdown. These amino acids join Kreb cycle as different intermediates. You can see. So you can see this. So this is a complete picture. Everything is incorporated into one picture. I did not find any picture like this. That is why I made like this. So you can say this is a cycle. So this is fed condition. So in fed condition, so glycolysis is facilitated. So this glucose will be broken down during fed condition. In fasting condition, glucose is formed. And glucose is formed, and what other pathway happens? Because you are in fasting condition, so fat is utilized as fuel. So from fat, energy is utilized. So here production of energy, and during gluconeogenesis, there is a requirement of energy. Okay, that is why, because it is uphill reaction, downhill reaction. Downhill reaction release energy, uphill reaction require energy. So that is why I told you uphill reaction. So how does it happen? This amino acids will join as intermediate of Kreb cycle. After breakdown of amino acid, they will join as intermediate of Kreb cycle. And this intermediate of Kreb cycle will ultimately form glucose. Remember that only. So the function of this Kreb cycle changes according to context. During fed context, the function of Kreb cycle is glucose oxidation, downhill reaction. During fasting condition, uphill reaction for synthesis of glucose. And this Kreb cycle is fed during fasting condition by amino acids. As you can see here, you can see this is Kreb cycle. So during this is pathway, you can see it is going continuously, but during fed condition, what will happen? This is the pathway. It is broken cycle during fasting condition. So this amino acid will join as this. These all amino acid join as succinate. This all amino acid will join as fumarate. This all amino acid will join as oxaloacetate, and through this pathway it will form oxaloacetate, and oxaloacetate will form glucose. So this is the gluconeogenetic pathway. We will talk in detail in MD2. So for the timing, just remember during fasting condition, purpose of Kreb cycle changes. In fed condition, Kreb cycle is for breaking the glucose. In fasting condition, forming the glucose. Breaking the glucose, it helps in fasting condition. It helps in breaking the glucose. In fasting condition, forming the glucose. So I have also given you mnemonics. You can use this. This is actually very important slide, but because I will teach you in detail in MD2, that is why I did not talk much about this. Just I wanted to mention that this is the function of Kreb cycle during fasting condition.